So here you hear me well? Okay. So hi everyone, I'm Tatiana, I'm a postdoc in Magali Suzanne's group. And today I'm going to talk about the impact of external constraint on cell and tissue dynamics. So in the lab, we are studying morphogenesis and how epithelium sheets undergo extensive reorganization and how they go from 2D flat tissues to 3D complex shapes. And one really good mechanism to do that is folding. And to fold, a tissue can undergo collective cell reorganization and undergo apical constriction on some cells of the tissues. And this has been extensively studied, but you can have also different mechanisms, such as differential strains between two tissues. So if you have two tissues that are linked to each other and one is growing faster than the other, then it's going to create folding. And this is what is happening in the gut, for example. In my project, I am studying the Drosophila leg disc. So it's an imaginal disc, and I can culture it ex vivo for several hours, which is really handy. And during this time, we are going to see the growth and the folding of this tissue. So when we look at it at the stage we are studying, you have already three folds that are formed already, and we're going to be able to fully visualize the last one, the last fold. And by doing so, the team identified a new folding mechanism that involves apoptosis some years ago. So when a cell is dying, it's generating an apicobasal force through an apoptotic myosin cable. And this is going to lead to the recruitment in the neighboring cells of myosin. And as the cells are organized along a pattern, which is genetically delimited, apoptosis is going to propagate, which is leading to the accumulation of myosin along this pattern, and which is leading eventually to the fold formation. If you look at the timing of the process, you have during the first 6 hours of ex vivo culture the last fold formation, as you can see here, so it's this fold, and then you have the removal of an envelope which is wrapping the leg. And the question of my project is to ask the role of this envelope. So first, is it exerting a mechanical constraint? And if so, how is it important in the fold formation? So to understand the role of this envelope, I developed a protocol to remove it. So here you can see the legs without the this constraint, without this envelope. And you can see that they have a really different phenotype when you compare it to the control ones. And in order to understand a bit more what has happening, I decided to quantify. So I measured the distal length of the leg. So at 3 different timing. So at the beginning of the process, at the middle of the process, and at the end of the process where the fold is supposed to be formed. And so what you can see is that you have you have 2 phases. You have a first rapid elongation of the leg within the first 20 minutes, and then you have a slower elongation phase. So with this measurement, what I can say is that the envelope is exerting a mechanical constraint on the leg. And then I looked at the morphogenesis. So I looked at the fold formation, and I measured the difference between what we call the segment here and the fold here. And I could show that at the end of the process, where the fold is supposed to be formed, you have a high defect in the fold formation. So we just show that the external constraint is important, is necessary for the fold formation. And on the other hand, what I told you before is that the apoptosis is necessary for the fold formation. So the question that comes from that is that if the external constraint has a role on apoptosis, and if maybe that would be the pathway and it could be regulating apoptosis. So this is the question I asked next. And to do that, together with Martin Casales, we developed another protocol in order to increase the constraint applied on the leg, and we took advantage on the fact that the leg is a growing organ. So we grew them in an agarose medium, and we chose the concentration on agarose to balance the force of the growing leg. As you can see here, so the well is developing well during the fold formation process. And with these three conditions, we monitor the apoptosis. So to do that, we have an apoptosis sensor that have been put into flies by the team. So we are going to be able to see the caspase activation, and we are going to induce it in a well-delimited region of the leg. And what we are going to do is to count the columnar cells, which correspond to the beginning of the apoptotic process. So the new apoptotic cells that are formed, the new cells that are dying, and we are not counting the apoptotic bodies that correspond to cells that died before, a long time ago. And what you can see is that you have this pattern that had already been shown. So you have the number of apoptotic cells through time. And when you grow the leg with more constraint, what you can see is that you have more apoptotic cells. And when you release the constraint, when you remove the envelope, you have less apoptotic cells. So what we just what we can conclude is that the external constraint is regulating the apoptosis, which is necessary for the fold formation. And what I asked after that is how could that happen? What is the pathway? How this is regulated? And so I'm wondering what are the physical parameters that are modified by the external constraint that could regulate apoptosis. And so we are going into the part of the talk a bit more exploratory. So I have a bunch of candidates, and we are going to test these candidates together. So first, I looked at the geometry of the cells. So I realized I performed single-cell staining to check the volume and the height. And what you can see, for example, is that the volume is not significantly modified, so it's probably not a parameter that regulates the mechanosensitive apoptosis. However, you have a difference in the height, and I'm especially focusing on the fold because it is where the apoptosis happens, so I want to see if there is differences in this region. And so this is a parameter we can keep and test later on. Then I wanted to have a look on the what was happening on the apical part of the tissue, but we needed a tool that was able to perform segmentation in really highly deformed tissues. And so we had to develop our own tool. So it is called Dissect, and it is now available for the community. And with this tool, we could show that the apical cell area is not modified. However, when you look at the anisotropy of the cell in the folding region, you can see that here, in the case of the constrained legs, you have a long tail of distribution that you lose here. So the cells here are way more isotropic. So it could be also something we need to check to see if it is regulating the mechanosensitive apoptosis. Okay, so this is something a bit different. So it's not a physical parameter, but it's something I came across while I was doing some experiments. And I called this the tissue's reactivity, but I could have called that tissue dynamics. When I was doing laser ablation, and I could not see any difference in the recall between the constrained and the unconstrained leg. However, and I think I need to touch. When I look at longer time scale, what you can see here is that when you do laser ablation at the level of the adherent junction, you have, in 80% of the cases, in the constraint leg, you have a strong process of healing, which is leading to the disappearance of one of the two cells. And weirdly, it's something that does not happen in the case of the not constrained leg. So you have what I would have called a normal healing, but apparently, well, anyway, so in 80% of the cases, you have just the healing of the junction, but you don't have any cell that does disappear. So it seems that the tissue here is way more reactive and dynamic. And similarly, if you perform ablation in the lateral part of the tissue, so here you have the apical part and here you have the basal part, you have, during the healing process, a strong contraction of the cells, while you can't see it when you do the same kind of ablation in the unconstrained leg. And this is summarized here. And so this is something I was not expected, and so I called that tissue's reactivity. Maybe that is a better word to call it, but it's the way the tissue reacts after a stimulation, or here an invasive ablation. And this raised for me the question of the dynamics of the myosin. Is it impacted? So what I did is that I did FRAP. So I bleached the myosin on the adherent junction, as you can see here, and I looked at the recovery of the myosin. And in the case of the constraint leg, what you can see is that for both of the conditions, the speed, the dynamics of the myosin is not modified. However, the mobile fraction is higher in the case without the constraint. So one hypothesis is that the myosin is less stable and the turnover is higher, so which could explain what we have just seen earlier, that the tissue is less reactive and is not able to contract as much as with the constraint. Okay. And so what I have checked as well is the membrane tension. So I did that with a probe, which is called the Flipper TR probe. So it's a probe that inserts into the lipidic membranes, and according to the membrane of the tension, it's going to change conformation. And this conformation can be reported by fluorescence lifetime. So here are two images. It's not a number of photons, it's the lifetime of the photon. And what I could see is that when you look again at the fold region in the apical part of the cells, you don't have so much change in the tension. However, when you look at the lateral tension, you have an important decrease in the under constraint leg. And when you look at the basal tension, you have an increase this time. And so this is something we got quite interested in, and we want to test if this could be a parameter which is important in the regulation of the mechanosensitive apoptosis. So to conclude, we've seen that the external envelope exerts a compressive stress on the leg disc, that this stress, this constraint is necessary for a proper fold formation, and that it does induces apoptosis. And together we've been through some of the parameters that could be involved in this mechanosensitive pathway. There are still some parameters I want to check. This I will do with the dissect software we've designed, and I also want to check the rheology of the cells, the cytoplasm. And I just want to emphasize that the leg disc, to do this kind of experiment, is a great model, because when you culture ex vivo model, it's kind of easy to apply a constraint, but it's quite difficult to release a natural constraint. And here we can do that. And I want to thank you all for your attention. I want to thank the people that work on the Dissect software, the people that work for me on the experiment, and especially Martin Casales, who's been a key actor in this project. Thank you.
Thank you very much.